This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 54 for October 16th, 2009. Hi, everybody. It's Vincent Racaniello, and today I'm in Princeton, New Jersey, at Princeton University with an old friend of mine, Lynn Enquist. Welcome, Lynn.
Good to see you again, Vince.
I've been waiting to get you on TWiV for a long time. Lynn is professor and chair of the Department of Molecular Biology here at Princeton. Probably the busiest virologist in the world. This guy is so busy, he does too many things besides being chair. You're the editor-in-chief of Journal of Virology.
That's right.
And a billion other things. So I'm lucky to get an hour from you on a Friday afternoon. I appreciate it. But this is a good guy to talk about viruses. And that's what we're going to do today. We're going to just chat about the things that you and I like a lot, besides fish.
Yes.
Or you like fish.
I like fishing. Fly fishing is my passion.
My colleague, Dick Despommier, who's not here, loves that.
I just sent you to show Dick a picture of fish that I caught in Chile this summer.
Because Dick sent, did you see
beautiful
the picture?
striper, yeah.
The striped bass he caught
yeah.
near JFK Airport.
Yeah.
I didn't know you could fish there.
Oh, yeah.
So next summer, we're going to do this TWiV in front of ASV audience.
Oh, yeah?
It's in Montana.
Yes.
So if you're there,
Yep.
because Dick's coming because he wants to fish.
Yep, I'll do my best to be there, too.
We'll get together again, and maybe you guys can show me what this fishing thing is all about, because for some reason, virologists like to fish.
Yeah.
I wanted to start by tracking your early days, because I know you starting at a small company in Minnesota, Molecular Genetics, but how did you get into virology? You obviously got into it before that, right?
Oh, yes, absolutely. No, I got my my undergraduate degree at South Dakota State University in bacteriology. I took food, dairy, water, sewage, all of the different kinds of bacteriology, and I began to get interested in viruses there because they were using viruses to type the bacteria that were used to make cheese. So different lactobacilli were typed by whether they were susceptible to different kinds of viruses or not. There were no virologists in the department at South Dakota State. Nobody knew much about it, and so I just kind of parked that away. And then I went to graduate school, started at the University of Minnesota in microbiology, and I took a course in molecular genetics, and it was taught by a guy named Peter Snewstead. Snewstead was a T4 phage guy, and so this was in the early 1960s. And early 1970s, late 1960s. And the molecular biology story was just unrolling. The Crick and Watson and Brenner and all of the stories of the genetic code and all of this. And I just found it all fascinating because the major workhorse were phages,
right?
Bacterial viruses.
T4 was a
T4
DNA phage. Right?
And Lambda, and which is another DNA phage. And Lambda had this property of when it could enter a cell and shut itself off and come back to life. And so I found this all just really fascinating.
We did phages then because they were the easiest to work with.
They were easy to work with, and they worked fast.
Right.
I mean, you basically could do 2 or 3 experiments a day if you were good at different kinds of things.
Great.
And so
a little different from herpes. Though.
Yeah, absolutely. Yeah, absolutely. So I was in graduate school for 2 years, and my advisor got offered an opportunity to be chair of microbiology at Medical College of Virginia in Richmond, Virginia. And he asked me to go along with him. Now, you have to remember, I grew up in South Dakota, and my parents didn't go to college. Nobody went to college. I actually had never been anywhere before. Minneapolis was the biggest city that I went to. On my college trip, we took the train to Chicago, and that was like the biggest thing in the world. So I jumped on the opportunity to go east. And I had a little interest in Civil War history, and Richmond was kind of in my mind. And so I went there, and I got my degree in microbiology. And the very last thing that I did after I got my PhD is I worked over the summer with a guy named Bob Kolstad, who worked on phage P22, a salmonella phage. And there I did all of the stuff that I had been reading about, making plaques, making mutants, doing complementation, recombination, different things. And I was really fascinated by what you could do with viruses. And I was looking for a postdoc, and I initially had the idea that I wanted to work on, of all things, potassium transport in E. coli. And the guy that did this is a guy named Herb Ennis, and he was at Roche Institute of Molecular Biology. And
It's
so,
up here in New
yeah,
Jersey,
right up here in
right?
Nutley, New Jersey. And I called, or my advisor called him or whatever, and he said he wasn't sure he was going to have a slot or not. This was the first year of their postdoc program. So I went up to interview him, and he arranged for me to interview another person, a woman by the name of Ann Skalka, who had just come to Roche from Al Hershey's lab, the guy who won the Nobel Prize for molecular biology and working on phages and things. And I was so impressed with her, I just couldn't believe it. And I just instantly switched, my allegiance right away from E. coli to Lambda that she was working on. And she took me. I was stunned that she would bring me in because I had really no experience.
Smart woman.
And she was really good at that time with the state of the art, which is what fascinated me about Lambda it was the paradigm for all kinds of molecular genetics for the whole business. If you could do something in Lambda, it was just amazing. Discovery of and the verification of things like repressors and operators and whatever was really being done, both in E. coli and the lac operon and in Lambda. And Anne was very interested in replication. And she convinced me that this was something that I should learn to do. So we did some of the very earliest experiments showing the interaction of recombination enzymes and replication enzymes in Lambda. And it was very complicated genetics, but suffice it to say that I learned the art of pulse chase experiments with radioactive P32. You would add P32 for 10 seconds, and then would add cold P32. I mean, it was very rapid, and these beautiful experiments. We'd run sucrose gradients to separate the replicating DNA, which was small and P32 labeled, the incoming DNA from the replicating DNA, which was tritium labeled and would get bigger and would be these long concatemers of DNA. And we realized that when the recombination systems weren't working, the DNA was shorter, and sometimes, depending on the mutations, they were circles. And Anne and I spent a lot of time putting this together. And with Anne's help, we came up with a model for Lambda switching from early replication, which was circles, to late replication, which was these long linear things that were packaged into into capsids. And we discovered that there were E. coli enzymes that were involved in recombination that regulated that step. And that model was around for a long time. Now, remember, I went to Cold Spring Harbor to give my first talk, and it was the Cold Spring Harbor phage meeting. There's a so-called Lambda book meeting in which the first book on phage Lambda ever came out. This was
when Max Delbruck was still going?
Yep.
And all the famous phage people,
So when I was getting ready to put this talk together, I was very confident in the whole business, but I'd never ever given a big talk before. And Anne took me aside and said, you're my second postdoc, and this is very important, and this is sort of groundbreaking work. People are going to ask you lots of questions. No problem. I don't know at all. I was just completely naive. If you ever been to Cold Spring Harbor before it was all renovated, in Blackford Hall, there was this, there's this picture of Watson standing up pointing to the
double
helix. The same stage was there. The same really long, flexible bamboo pointer was there. And so I got up to give my talk, and all of a sudden it dawned on me that this was where it all happened. And the stage was very narrow. And I got up on the stage, and I looked out in the front row. It was Al Hershey was sitting there, and Delbruck was there. Mark Tashney was there. David Botstein was a postdoc at that time. Maybe he was a beginning assistant professor. He was pacing in the back of the room.
Was Sal Luria there?
I didn't see Luria. But all of a sudden it dawned on me, oh my God, now what am I going to do? So I gave my talk, and afterwards Frank Stahl came up to me. And of course, he was one of my idols, the Messel and Stahl experiment.
Right.
I thought he'd be a really old guy, but he was a fairly young guy, very tall. And he invited me to play ping pong. We played a lot of ping pong in those days, and he was really good. He just beat the bejesus out of me. And then we sat down and he talked. He says, "This model is really great." And he went on to show how he was working on very similar things. And I was sort of blown away by this. Well, first of all, as a kid from South Dakota, people were listening to me that were my idols, and so it was a big deal. And then I met these guys from the NIH, the Lambda group, Bob Weisberg, Max Gottesman, Marty Gellert, Howard Nash, all of these really big name guys. And they listened to me, and I found that to be astounding. And I got an opportunity to go to the NIH after I spent 2 years at Roche.
So you were a postdoc at Roche,
At Roche,
right?
yeah, for 2 years, and then I got an offer to be what's called a staff fellow at the NIH to work with Bob Weisberg, who was in Phil Leader's big lab group. And Phil was a famous geneticist, and actually he had worked with I think it was Nuremberg, but they sort of deciphered the genetic code with triplet binding assays and things like that. So once again, I was falling into this really famous collection of people.
Do you feel chained to your office computer? You have everything on it. You can't go anywhere. You can't even go home at night. Well, here's a solution. My recommendation: break the chains and use GoToMyPC. You can access your office computer from home or anywhere. No more late nights or weekends in the office. All you need is an internet connection. Use any program, work on any file, check your email, access your network resources. Really easy to set up and as secure as online banking. All you have to do is go to their website. You'll get a login and a password, and then from any internet-connected computer in the world, you can access your own computer in your office. Now, our listeners, the listeners of TWiV, can try GoToMyPC free for 30 days. That's a month of unlimited remote access for free. For this special offer, you must visit www.gotomypc.com/podcast. That's gotomypc.com/podcast for a free 30-day trial. We thank Citrix for their support of TWiV.
I got a little distracted at that time because the Vietnam War was going on, and at South Dakota State, everybody had to join ROTC, and I did. And because we didn't really have a lot of money, I went on and got my commission in ROTC because I got a uniform and I got $200 a month or something like that. And I got called up to active duty after my first postdoc, and I was really kind of spooked at that because at that time I was an officer, and the half-life of officers in Vietnam was really low, and wasn't so sure that I wanted to do that, but I didn't know anything else to do. And so I called up Phil Leader and said, Phil, I can't, you're going to have to delay me coming, or I'm going to have to turn it down because I just got called up in the Army. And he says, well, I can get you in the public health service. I have a slot for a public health service officer. I didn't realize public health service was the same thing as, the Army, Navy, and whatever. I says, you can do that? And he said, yeah, send me your particulars and we'll pull it off because these guys really want you there. So 30 days later, I got an official letter from the Army indicating that I had been discharged from the U.S. Army, and I was now appointed as a lieutenant commander in the public health service, and I had to report to Bethesda at such and such a time, and so that was great.
Why is the public health service and the Army together like that? I never understood
I have
that.
never understood that, but it was part of the idea that there was a need, I think, for the government to take care of people, particularly Native Americans, people that had leprosy. There were a few other things that the public health service really had to take care of.
Yeah.
And if you look at the history of it, it's kind of interesting. But at that time, to me, I was very happy that I could continue with my career.
So that allowed you to go to Weisberg's lab.
That's right.
And
that's right.
And you worked on phage?
I worked on phage. I worked on lambda there. And there I really kicked into high gear in pretty deep lambda genetics. We studied site-specific recombination, the mechanism by which lambda DNA inserts its DNA into the E. coli chromosome and how it comes out. I mean, really sophisticated genetics with very little biochemistry. We used to always laugh that we could do everything with our brains and toothpicks and didn't need any of those restriction enzymes. In fact, there weren't restriction enzymes then.
Not yet, right. So you were studying, and others were studying, integration of lambda DNA into the host.
Yep.
It was before we knew about retroviral integration, right?
Yeah. It was the only paradigm in town, and that paradigm, which was developed by Alan Campbell, who genetically showed that the phage, the two ends of the phage, the markers on the ends, joined together when the phage was replicating, and when the phage integrated, these markers actually were in the middle, and the chromosome was split at a different place. So he called those attachment sites. And when lambda integrated, it always went between the gal and bio-operons. That was its primary place in E. coli. And so there was a lot of tricks that you could do with lambda in those days. It was so much fun. One of the things that Bob got me interested in was the idea of making lambda integrate somewhere else. And at that time, they had already known that there were places called secondary attachment sites. If you deleted the sequence between gal and bio, which you could do genetically, no restriction enzymes required, you still could get lambda lysogens. And when you looked at them, they were in different places. And you could identify these genetically because occasionally lambda would pick up a piece of DNA from the left or the right side. And if you were good at genetics, you could show that it was a transducing phage, and it would recombine with some mutants that you had. So you could say it was lambda in proline or lambda in, in the trip operon or something like that.
So probably we should explain that a transducing phage is picking up a piece of E. coli DNA.
Yes, and that's because the process of excision is precise up to a point. And if you know how to look, you can find that there are rare times that the phage makes a mistake and comes out carrying a piece of DNA either to the left or the right to where it went in.
Of course, nowadays we can just sequence it, but back then you had to use genetics.
Yeah, we used genetics. We didn't have sequencing. There was no restriction enzymes or anything. And I learned all these tricks from these guys. And it used to be a real challenge to design an experiment for which maybe you'd only get one plaque or one colony, but it would prove that you actually had what you were looking at. So what Bob set me on was, first of all, to find secondary attachment sites and map them. And I started that project. I didn't have really good ideas of how to do that. But one of the things that Bob also showed me was that there was a place where lambda integrated into the gal operon, the synthesis of galactose. And when it did so, the bacteria became gal minus. And I found out that when lambda excised from that, it did so precisely, and the cells became gal plus again. And so we had an indicator dye, so that when you made a lawn of this gal minus bacteria on this plate that had an indicator dye on it, you could see when lambda excised, there would be red cells in the middle
gal
of the
plus
in the middle of the plaque. And then it hit me right away that this was a screen for integration and excision mutants.
Mm-hmm.
So that if I plated a mutant that couldn't integrate or a mutant that couldn't excise, it would not make red plaques.
Right.
And so I isolated thousands of int and excise mutants, and I mapped them. I made deletions. I mapped them all across the lambda chromosome. I found exactly where the int gene and the excise gene were. We had good ideas of where they were, but I had enough mutants that we actually could figure out about how big these genes were. And I figured out that the int gene was, sort of standard size, fairly big, but excise was really small. The gene that encoded the protein that excised it was tiny.
And that's where the mutations would have been.
That's right.
That's right.
And so, and then I found a mutation that was both integrase defective as well as excisionase defective, and it mapped right at the junction between the two genes, and so I deduced that the genes were overlapping.
Huh.
And so we had all of these things. I also found temperature-sensitive mutations. I found some mutations, for example, that were in the integrase gene that actually could integrate perfectly fine, but they couldn't come out.
Mm-hmm.
And so that said that int protein and excise protein were in a complex, and that we were screwing up the complex. We were figuring all of this out just by whether you made a red plaque or not.
It's amazing.
And then there was another guy who was really sort of my genetic idol at that time. His name was Mark Schulman. He was at the NIH as well. He was a postdoc, and he was doing genetics of the attachment site. And so the attachment, that's where lambda integrates.
Integrates into E.
coli. So there's a phage attachment site and a bacterial attachment site.
And it's always the same place, more or less.
It's the same place, more or less.
Not like retroviruses.
No.
Right.
And he was isolating mutations in lambda in the attachment site, and he could prove that they were there because they didn't affect a protein. They were so-called cis-acting mutations. They only worked on the DNA that had them in. And we knew it was very close to the int gene, and we had all of the mutants, so we could tell that they were there. And you discovered that there were some interesting properties. So if you integrated a phage that had an attachment site mutation into E. coli and then popped the phage back out, you discover that, and you could go back and forth, and you could figure out whether you could look at the left side of the integrated virus or the right side, and the mutation would move from one place to the other.
Mm-hmm.
And from that, we deduced that there was a common core in the attachment site, and there must be staggered nicks so that you got essentially the gene moving to the
all
other
without
side.
sequencing.
All without sequencing.
And then at some point later on, somebody did.
Art Landy got the sequence,
Yeah.
and I remember when we got the sequence, we looked at it and we said, well, we knew that.
We knew that.
But it was too cool to have the attachment site, because then you could really see what went on. The other thing that I did with Bob Weisberg's help and Howard Nash's help is that I had done some crosses between two int mutations, and I did them in a E. coli rec minus cell. So E. coli didn't do any recombination.
Mm-hmm.
We fixed it so the phage didn't do any of its recombination. It could only do site-specific recombination. And we knew we had the attachment site here, and the int gene was there, and I had a phage that had an int mutation there, and then another phage had an int mutation there. They were separated. I knew they were separated. They could be recombined. And I got int-promoted recombination between these two guys, away from the attachment site.
Mm-hmm.
And I was showing this to Bob, and Bob said, you know what that might mean? That might be telling you about the mechanism of recombination. The idea was that what you got is what we would call a holiday structure. So basically, there was a crossover at the attachment site, and the crossover actually moved into the int gene, and then it was cut, and you got recombination. So we have a paper that basically said that the lambda site-specific recombination mechanism involved a holiday structure. It wasn't, just cutting and pasting and whatever. And we did it essentially by a process that we call was branch migration, where the holiday structure moved.
So the holiday structure, the two different DNAs are swapping in the parts that they're sharing with each other.
They're sharing,
right?
But in fact, so one strand goes into the other one, the complementary strand goes in the other.
It can move.
And then that crossover moves.
Yeah.
It's like taking two, cables and twisting them. And if you have a cross between them, it moves up and down and moves down.
Yeah. I'll have to put a picture in this episode to show people, because it's a little, for people that are not used to thinking about it, it's a little abstract.
Yeah. But it turns out that at that time there were a lot of people interested in just the mechanism of recombination, and that we were thinking site-specific recombination was different from homologous recombination, different enzymes were involved, but the intermediates all started looking like they were alike. So I could go to recombination meetings and talk about our site-specific work and be talking to people who were just really right on top of it and making sense of it all.
So at what point did the holiday recombination ideas go over to eukaryotic cells? After that?
Well, it's pretty much after that, because at this time another thing began to happen. So of course there were eukaryotic genetics going on. When you do certain crosses with different kinds of mutations, and some of them were done in fungi where you got spores, and other of them were in different cases. You got what were called sort of aberrant recombinants, aberrant tetrads and whatever. Instead of getting equal numbers of the parents in the offspring, you got weird numbers, 5 and 3 and 6 and 2 and things like that. And the idea was that somehow you got a recombination event in one strand, and then it moved, and you finished the recombination in another. And so they were talking about, holiday structures and things. A guy named Robin Holliday was thinking about these.
That's
where the name came after. Yeah, he is from England.
So this is a great example of how phage set the early molecular biology, just because the genetics was so easy.
The genetics was there, and then people would say, hey, that looks just like what we've got.
Yeah.
And then they would come up with a model or some ideas, and we would test them in bacteria. And there were a lot of people working on this problem. So it was a great community to be around. And at this time, because we were thinking about recombination and whatever, there was another group of phage folks and bacterial folks that were studying another problem, which was exclusion or restriction. That if you grew viruses on one kind of bacteriophage and then you plated them on another one, they wouldn't grow. But if you looked close enough, you would find that a few would grow, and you'd pick these guys, and now they would grow all the time. And there was a group in Switzerland, a group in England, a group in the United States, and it was discovered that the bacteria made enzymes that they could mark their DNA. So they know everybody who grew in there had a special mark. It was a methyl group in certain places. And they also had enzymes that recognized that DNA sequence, and if the methyl group wasn't there, they cut it. And it was sort of primitive immunology. It's
right.
A virus,
innate immunology.
If a virus came in that didn't have the methyl group in the right place, it was killed.
Yep.
And
it was called restriction,
restriction. Right? It was called restriction because it restricted the replication. And for a while, we were just using this as tools. Just the fact that you grow a phage on one kind of E. coli and it wouldn't grow on another was just another marker, and we weren't thinking about it that much. There was different kinds of restriction enzymes and different things, and we started thinking a little bit more about what the value of this stuff. And some work was being done in trying to make transducing phages that carried different DNA, E. coli DNA and whatever, using enzymes. So instead of having E. coli do it for you, you actually purify the DNA, and you added some sequences to that. When I was at Roche with Ann Skalka, I was next door to a guy who was working on silkworms. His name was Pat Gage. And Pat was really keen on the silk gene. He wanted to clone the silk gene. But this was before cloning, before restriction enzymes and whatever. And there was a guy out in California at Stanford. His name was Peter Loban. And he worked with Dale Kaiser, who was a phage geneticist,
right.
really famous phage geneticist. And Loban was working with P22, and he found out that you could stitch molecules of P22 together, and the idea that maybe we could stick DNA on the ends was one thing. There was a big group at Roche that sequenced the ends of, that synthesized the ends of lambda that were involved in circularizing the DNA. The idea that you could put those ends on any DNA and they would circularize and maybe lambda would pick it up. There was a lot of this going on in the early 1970s about how can we hook DNA onto something that's replicating. Nobody was thinking of recombinant DNA or things you think about. It was just a way of studying recombination and figuring out. Yeah. And Anne and I did some experiments that had to solve two problems. One is how could we get DNA hooked onto lambda? Mm-hmm. So we knew that lambda picked up gal and bio and other kinds of DNA when it excised, so it told us the place that we should be putting stuff. And we tried to figure out how we could clone things into there. We didn't use the word cloning because that was not a word at that time.
Right.
But Anne had the idea that we could use poly A and poly T. So and they're complementary base pairs. And so if we put single-stranded A's on the ends of one molecule and put single-stranded T's on lambda and mix them together, they would join up.
Right.
That was cool. We could actually do that. But the next problem was getting that back into E. coli again.
Right.
And that was hard. Nobody had really a good method for getting DNA.
But we knew that you could put DNA in E. coli because Avery, McLeod, and McCarthy had done it.
Yes, with Pseudomonas, or was it Pseudomonas or Pneumococcus?
Pneumococcus in the 40s. But it was probably very crude and it hadn't been developed very much yet, right?
Yeah, there was a guy named Morton Mandel. I think he was from the University of Hawaii. He might have been at Rockefeller when he did this. He showed that if you treated E. coli, I think, with calcium chloride or something like that, it would take up DNA. We tried that, and that was just not a very good technique. But there was a guy down in University of Virginia. His name was Rolf Benzinger. Had developed this assay in which you took the cell wall off of E. coli and made spiroplasts.
Right.
And then you would add DNA in a little osmotic shock and would take it up and you could plate it out.
Yeah.
So there were a number of people using spiroplasts to put DNA in, and there were a variety of other groups that had shown that you could add DNA, lambda DNA that had the gal operon in it, and then if you infected with bacteria with lambda, it could help the DNA in. I think Dale Kaiser and a few other people were doing it. So there was a lot of stuff that was going on so that we could get DNA and viruses and lambda into a cell and get new viruses back up. And all of this was happening, sort of at that time. So when I got to the NIH, I was really primed for what was then the revolution that Jackson, Simons, and Berg showed that you could combine SV40 and lambda and get them together in the same molecule using these restriction enzymes that these guys had discovered.
Right.
Because the restriction enzymes, when they always cut at the same sequence, and then you could stitch them back together again. And so I got involved sort of as a secondary project of developing lambda strains that were really good for picking up DNA and making E. coli strains that were high efficiency transformation. And Nat Sternberg, another postdoc in the lab with me at the time, Nat was studying the process of lambda assembly in vitro. So I don't know how many genes are involved, 20 or 30 genes. And you had all of the proteins in test tubes and mixing them together, and we called it in vitro packaging. You'd build a particle, you'd add DNA, get it picked up, and you could get it out. And so Nat and I worked out ways to make this really efficient so that you didn't have to do spheroplasts and all this wacky stuff. You could just mix it all up in a test tube.
It's built to put DNA
That's right.
in.
That's right. It was and so that was really sort of a closet industry. And I met a lot of people then who said, "Will you clone this gene for me, this gene for that gene?"
At NIH.
At NIH. And so the lab that I was in, Phil Leader was the big lab chief. And in his lab were several outstanding postdocs. One of them was Shirley Tillman. She's now my boss. She's the president of Princeton University. John Seidman, who's now at Harvard with his wife. There's another guy named Peter Tiemeyer was there. There's a couple of other people whose names escape me right at the moment. But Phil wanted to clone the globin gene. He wanted to get the human, well, I guess it was the mouse gene, mouse globin gene out and get it so that we could study how eukaryotic promoters worked and all of that. And so we worked really hard. I helped them. I made the phages. We developed different kinds of assays. It was really tedious. We didn't have any of the fancy stuff to do.
No PCR, no sequencing.
No, nothing. No
oligonucleotide primers. Yeah.
We had something called the gene machine, which is a humongous gel electrophoresis thing that would separate fragments. There was a column material we used called RPC5. It had been used in countercurrent distribution of transfer RNA, purification of transfer RNA. It separated DNA by magical things, and we could show by the combination of gel electrophoresis and this RPC5 column chromatography, we'd get 1,000-fold enrichment of certain kinds of restriction fragments. And then we clone from that. And we'd pick 5,000, 6,000, 7,000 plaques and screen them by hybridization to see if they'd picked up the globin gene. And one weekend, Shirley was in the lab and found the first globin clone. And so she grew it up. I can't remember if I helped her or not in that, but they started looking at it, at what the gene looked like. And to look at it, they had some messenger RNA of globin. So they had the purified messenger RNA. And there was a technique called R-looping, in which you would take the purified DNA fragment, and you would hybridize the RNA that had been derived from it to that fragment. And if you looked in the electron microscope under proper conditions, you could actually see a loop. The double-strand DNA RNA plus the loop of the DNA that was sticking out. So I remember going down into the EM the first time to look at this thing, and it wasn't the simple loop that we thought we would see, the double strand with the loop out. The double strand had loops in it, as if something had been taken out of the out of the message, and they were very precise. The right side, there was 2 of them, and at that time, I'd also become very interested in transposons. So little pieces of DNA that jump around and whatever, and they have very interesting genetic effects. And I thought, oh my God, what's happened here is this globin gene has been cloned into E. coli. E. coli has never seen it before, and it's putting in a transposon in there to shut it off. And I remember talking to Phil about that, and he was rolling his eyes saying, "You're crazy, it can't be." And I said, well, it's really easy to figure this out, Phil. We'll just get a couple more clones. And if they're all in the same site, then it's a biological thing that's interesting.
Sure.
And if they're not, then it probably is a transposon. Well, we didn't know it at the time, but we were looking at splicing.
Yeah.
Maybe we're the first people to see it.
What year was that now?
So that had been 75, 76,
not
somewhere
too long
right
before it was.
Well, that was the summer of splicing. So that it was discovered, Cold Spring Harbor. There was Phil gave our talk at the Cold Spring Harbor meeting. At that same meeting, Robert Sharp and Broker presented their work on adenovirus, which showed
Phil Sharp,
Sharp, yeah,
right.
That showed that adenovirus late message had the same structure at the end of every message, and yet it was all splicing.
It was all by EM, right?
All by EM. And
wow.
So we missed the big thing. I hope it wasn't because I was trying to convince Phil that it was not a transposon. But we picked up a few more clones. They were in the same place.
And they were all the same. So when you were doing this initial cloning, did you have to do it under high containment?
No.
Because nobody had any clue.
There was no containment. There was, at the time, there was concern. There was the Asilomar meeting either was about to happen or had just happened. And if it had just, yes, I think it had happened, because Phil and I did a lot of work to show that the lambda vector that we used was, quote, safe. I had put some nonsense mutations in it, so it only grew in certain strains of E. coli. We had done a number of things to show that the probability of this thing, getting out and growing in the wild was really, really low.
That was the concern, that you would make something that would get out.
Yeah.
So Silomar was a bunch of scientists who got together and said, we're a little bit afraid about this, we're going to regulate it. Right?
Yeah, well, so this was a good example of scientists all of a sudden realizing that they had new information that they had to stop. They had to think about what it meant. Because you could see, well, all of a sudden, the species barriers were broken. Not only species barriers, but genus barriers. I mean, we could clone plant genes into E. coli. Sure. We could do almost anything. And the principle was so simple that anybody that basically was not too much of a klutz in the lab could do it. This was not stuff that could only be done in one place and whatever. Sure. So a lot of people got together. I was involved mainly from the NIH in helping to write rules and guidelines, and I worked with a group that really decided what kind of safety features you could build into vectors, that is the things that would carry the DNA, so that they would stay put. And we developed the concept, we being everybody, that you would have physical containment as well as biological containment.
Right.
So, and we had names for these things, but the combination of the two would mean that there were certain experiments that you could only do under the highest possible containment, essentially biosafety level 4, with a very special kind of vector that was so crippled that it would only grow under certain circumstances. And so all of these rules and regulations came out. But basically for the simple cloning of mammalian genes, I was involved in a number of these experiments just because I had the lambda phages, I had the in vitro packaging, we had the enzymes, we knew how to do it. So if you look at my CV at that period, there's 4 or 5 papers where we cloned genes that didn't seem to have anything to do with my interest in phages and
You're doing it for somebody, right?
Yeah, I basically did the work, and they were kind enough to put my name on the paper.
Weren't you doing some SV40 cloning at some point?
Yeah, so after I finished in my 4 years in Phil's lab, I was still in the public health service, so I had a job at the NIH. And George Vanderwood, who was a guy I just got to know and admire, was a retrovirologist. He also worked on foot and mouth disease virus, one of your favorite picornoviruses. and I met George, and I really liked him. He was just a sort of a down-home kind of guy, but he was in the Cancer Institute. And in the Cancer Institute at that time, there was a series of lab directors. There was George Coury, George Vanderwood, George Tadero, Bob Gallo, Ed Skolnick. There were all of these famous virologists, very powerful guys, using sort of doing state-of-the-art animal virology. No one was doing phage stuff anymore. And George Coury took me aside one day and says, "Yeah, you ought to come over and and to bring your recombination technology over, and you should work on real viruses."
Real viruses.
Yeah. I got suckered into that. But George was such an amazing man, and he worked on SV40. And the year or two that I knew George, he died way too early from lymphoma. But he had they sequenced SV40. They discovered enhancers and all of this stuff. It was just an amazing time to be around. And he was in the lab right next door to where my lab was, and George Vanderwood was my boss. And George convinced me to help them clone integrated retroviruses. So that's where I met you, actually, because this came about that we worked on MSV, murine sarcoma virus. And George had been working on this for some time, characterizing the virus particle, the proteins, and he desperately wanted to get a hold of this thing. It was a defective murine leukemia virus.
So it's an endogenous.
And it was a virus that they knew at that time, because the story was just developing from Bishop and Varmus and whatever, that these sarcoma viruses had picked up cellular oncogenes. And he wanted to know what the MSV oncogene was.
Right.
So at that time, no retrovirus had been cloned. And so we went after the integrated retrovirus. And I had to do this under biosafety level 4 conditions in Building 41 at the NIH. and so we took, I think it was integrated into quail cells. And so that we could make a probe, because there were no clones, the probes were made in very strange ways by hybridization and different kinds of
things. So the quail got rid of the mouse genome.
Yeah. And so we made the probe from mouse cells and then hybridized and the only thing we could see was the oncogene that was in there. So we cut the DNA up, we did the big gel in the RPC5 column, and we cut out the DNA. And George had a really magnificent technician named Marianne Oscarson, and we worked in these Class C cabinets wearing, green pajamas and the whole business. And we did this cleaning. We got it. And Marianne and George showed early on that you could just transfect that DNA into cells, and they transformed them. So they knew they had what they wanted.
Wow.
And there was also George Curry had a man named Ravi Dar who was a Maxim Gilbert sequencer in the early days of sequencing, where we used this old-fashioned sequencing. We sequenced the ends of the integrated virus. So we had cellular DNA, and then it turned into the retrovirus, and we sequenced those ends, and the ends were identical. It was the same sequence, and I can't remember how big it was, but it was a direct repeat. And we got very excited about looking at that. It was the first time, actually, anybody had ever seen an LTR, which is the long terminal repeat. And it was the promoter that drove the retrovirus. Well, at the same time, your old postdoc boss, David Baltimore, was doing the same thing with a different virus. And George Vanderwood, said, we've got to try to get this published quick. We contacted David, and I think we have back-to-back papers in PNAS or something like that. But we named the LTR the TRL, terminal repeat something. I can't remember, but it didn't flow off the tongue. And David named it the LTR,
and that's what stuck.
And so that was a neat experience for me. And it was the first time I met David Baltimore. And at the same time, Ann Skalka had switched from working on Lambda to work on Rauch sarcoma virus, studying integration. And she wanted to clone that virus, and the only place you could do that was at our place, because we had the only biosafety level 4. So she came down with a postdoc named Bill McClements, and I worked with Bill and Ann and NP4, and we cloned that virus. Then I was talking to George Curry. He says, well, you can't go on like this. You can't just be somebody's technician, and clone things. He said, why don't you, Vanderwood has always had an interest in herpes viruses, and why don't you clone those guys in E. coli and then study them? He said, they have a latent analytic pathway. It's just like Lambda. Maybe you could find a eukaryotic repressor. Maybe you could, there's all kinds of things. There's naked ground, man. There's nobody working on that. And so there's a lot of people working on herpes viruses, they said, but they're not doing anything like what you can do if you had it cloned. And I talked to George, and George says, that's fantastic. I've got this whole project on defective interfering particles. When you passage herpes viruses at high multiplicity, most any virus actually, do serial passages over and over, the infectivity drops, and a new thing shows up, and this new thing is an origin of replication in a packaging site. It was basically a defective genome that can outcompete everything else. And so, but he wanted to know what the structure was, because the DNA was huge. He said, how can it just replicates in packages and doesn't seem to have anything else? What's all that extra DNA? So I had a postdoc, my first postdoc at that time, and we cloned defective interfering particles, and we characterized them with restriction from herpes simplex virus. And we discovered that they were essentially iterations, 17 or 20 repeats of a sequence that had an origin of replication and had the packaging site. And it did this by weird recombination. I read those papers now, and I'm stunned we actually figured out how all this stuff happened. But I looked at these, and I realized, and we didn't do much with this, unfortunately. I wasn't smart enough. I was smart enough to see how to use them, but I didn't do it. If you took that clone fragment that had the packaging site and the origin in E. coli, grew it up, and you added that DNA to cells, and you infected with herpes virus, this thing replicated. It was the first eukaryotic
origin
vector.
Of replication.
It was a vector.
Yeah.
And we could clone stuff in it. Yeah. And we have a couple of papers that said you could do this.
Wow.
And but I didn't have any interest in doing that, so I kind of let that go by.
But that was your first introduction to herpes viruses.
Yeah, and then I got pretty excited about herpes viruses. Because at the same time of doing the defective interfering particles, we had cloned essentially the entire genome of herpes simplex virus as EcoR1 fragments, Hindi3 fragments, restriction fragments. And I made those available to the whole herpes community. Anybody who wanted could get them from me. And that endeared me to the herpes community because nobody else could clone them at that time. And I made a lot of friends, and I discovered there were so many things to work on. I cloned the first thymidine kinase gene, which French Anderson got right away for his gene therapy experiments. That I wasn't interested in gene therapy, but
which Axel and Silverstein
yeah, that's right. That's right.
That's
right. That's right. There were two guys that cloned herpes TK. Steve McKnight was at Johns Hopkins, Carnegie Institute in Baltimore, and me. And our two genes, one had a restrictions on a SAL cut site, and one didn't, and you could tell who got what. And Steve went on to use the TK gene to characterize what a eukaryotic promoter was all about. And I didn't do that either. So, I was interested in how the genes were organized, and I was looking for the repressor, and I was looking for other things, and I could never find anything that gave me any clues, and it was so hard compared to Lambda. This thing, Lambda made a plaque basically sort of overnight, and the replication cycle was done in an hour.
Yeah.
And the Simplex virus, it took like 24 hours for the replication cycle and maybe 2 or 3 days to make a plaque.
Yeah.
So I worked, I tried on that. And one of the things that I got that kind of opened up the next stage of my career was I cloned with a postdoc named Roger Watson a gene that when we did a little bit of sequencing, it had a novel sequence at the amino terminus. And at that time, Gunter Blobel and guys had just discovered and made the hypothesis, the signal sequence, that if you're a membrane protein, you had to have this special sequence on your amino terminus.
He hypothesized that, but he didn't have the data to show it.
He was collecting a lot of data to show that, and there were a lot of people at that time, I would say a lot, but a few people were beginning to realize that most of the viral membrane proteins and things that we were interested in might have signal sequences, but nobody had ever really seen one before. And in fact, nobody had ever cloned a membrane protein from herpes simplex virus at all. So my postdoc Roger Watson and I had this clone of this protein that looked like it was a membrane protein. And the only guys I knew nearby who worked on membrane proteins was Gary Cohen and Roz Eisenberg at the University of Pennsylvania. They were, 3 hours from the NIH. And so I had read some of their papers, and I said, Gary, I've got a membrane protein. I'm sure of it, but I don't know what it is. And they had been making antibodies and different things. And they had this membrane complex called CP1. It was a complex of a variety of different proteins, and they made antibodies to these proteins. And he came down to the NIH, and we did a few experiments in collaboration and whatever, and we had a very good idea that the gene that we had cloned was a protein called GD. GD turns out to be the ligand for the receptor.
Right.
And at that time, there was no HIV, and herpes simplex virus was like the scarlet letter. I mean, it was on Time magazine's cover and everything. It was a horrible infection to have, social disease. And I got to thinking, maybe this thing could be a vaccine, could be a protein for a vaccine. So in 1976, Genentech was formed. So this would have been in 78, 79, 80 when we started doing this. And a little company in Minneapolis and Minnetonka called me up, and I knew these guys. One of the founders was Tony Ferris. He was a retrovirologist, and he had known of our retrovirus work and whatever. He said, why don't you come and help me run this company, and you could bring your GD gene, and maybe we'll make a vaccine. And so his company was a vaccine making company. It turned out actually to also do corn genetics and a variety of things. And we raised a lot of money from American Cyanamid, Letterly Labs, to work on a herpes simplex virus vaccine. And we expressed the protein in E. coli. We made antibodies that neutralized the virus. We got a paper in Nature. We got a lot of press. I got a huge number of hits. Radio stations from Australia calling up, and all this for the herpes vaccine. Yeah. When's it going to happen? When's it going to be? And I got very good at saying, 10 or 15 years, it's going to be well. I wouldn't count on it. And it was my first taste of working in a little company. And I kind of liked it because they were really smart guys. They were all young men and women that were so hot about all of the new things we could do in biology. And we all wanted to do something useful.
Did you miss having all these giants at NIH around?
I did. It turned out that in about 3 years, I realized I was not having the same kind of intellectual fun as I did before. And I also, the title wasn't called research director, it was called executive scientist or something like that. But my main job was really to raise money and to try to help people keep their projects in line and whatever. And I didn't get a chance to do any work. And I had one of the postdocs who was with me at the NIH was there, and I hired a technician who actually was Bob Tijan's technician. Tij, very famous transcription guy, and his name was Alan Robbins, and Alan was working with me. And one of the projects I gave to Alan was to work on a virus called pseudorabies virus. We're going to make a vaccine against this virus, which is a big problem in pigs.
It's an agricultural problem.
Agricultural problem. And
so it has nothing to do with rabies, right?
No. I was just going to say that it is a herpes virus. It's related to Simplex virus. But unlike Simplex virus, this thing grows like a weed. Its replication cycle is single step is about 12 hours, and it makes plaques in a day less. Its DNA was highly infectious. We could transfect cells and get it. And I just fell in love with this thing as a model for herpes viruses, because there was all sorts of things to study in that. And this virus was just great. And the best thing was nobody was working on it. I didn't have all of the Simplex virus people breathing down my neck and whatever. And so I started doing more and more with this virus, sort of alone. There were 3 or 4 other labs in the world that worked on it. And I started having a lot more respect for the veterinary virologists. These guys were doing great work, studying real hardcore problems in immunology and pathogenesis and epidemiology and whatever, and they were sort of looked down on because, they weren't doing anything with the hot new science and all of that.
But now, of course, that's changed.
Swine
influenza.
Yeah. Well, and because of that, I met a whole new class of folks, that were involved in variety of things.
So molecular genetics let you work on it because it was an agricultural problem.
Yes.
Okay.
And so and we actually got quite a bit of money through contracts to make proteins and make clones and do things like that. So we were successful. But I had to do the things that interested me really sort of under the cover. Yeah. And Alan was just a magician in the lab. And all of a sudden, I realized I didn't want to do this. I really wanted to somehow get back to an NIH type environment. And also, I began to realize that business was really hard. I didn't know how smart business guys were, and I didn't know how much money really drove the show, even though money is required for everything that we do. But in business, if you don't make money, you're not going to work. And so the company began to be really worried about how much money we were actually going to have to run the show, how much money we had to raise. I raised, millions of dollars over a period of time. And we were burning it up like crazy. And in fact, the people who were giving us the money said, burn it up. You're not putting any of this in the bank. You got to get some work done for us. So at that time, Phil Leader, who was my ex-postdoc advisor, called me up and said, DuPont is putting together a big biological center. And they've recruited some people. They've recruited Mark Pearson. Mark was a phage guy that I knew from Toronto. They recruited Nat Sternberg, who was my postdoc buddy at the NIH. A bunch of people from Nat's lab. A variety of people were coming to DuPont, essentially to do basic research on fundamental problems that, like Bell Labs, would have some practical things down the road.
Sure.
And it was called Corporate Research and Development, CR&D. And so I left molecular genetics and I went there.
That was in Delaware.
And that was in Delaware. Moved my family to Delaware. My wife said, don't you ever move again? Because we had been doing a lot of moving around. And DuPont was fantastic. It was exactly what they said. We got the freedom to do whatever we wanted. I had unlimited resources. At that time, the postdoc that I worked with in Bob Weisberg's lab, Nat Sternberg, was really bringing to fruition one of the things he discovered in Bob's lab and when he worked at Frederick afterwards, which was he discovered Cree. Cree recombination system, the LOX sites that Cree recombine, which is used all over the place.
System.
So he brought all of that to DuPont, and they basically put it all together. They showed that they could make it work in yeast, that it would work in plants, that it would work anywhere, and it was a great time. I was developing the concept. Now we would call viral vectors, but we called it viral engineering, which was to make viruses do things for us that would be practical. Bring viruses into cells to change the phenotype, so that if you had an assay for a ion channel or something like that, and it only worked in strange kinds of cells, we would clone it, put it into a virus, move it into another cell that you could really manipulate and do things.
What viruses were you using? Pseudorabies?
So we're pseudorabies virus, herpes simplex virus. There was another group that was interested in adenoviruses and whatever, pox viruses. And so we got that started. And then DuPont all of a sudden just decided that they were going to do away with basic research, and they were going to get more stockholder equity. We're going to spend more money on developing products. And so they formed a joint venture with Merck called DuPont Merck Pharmaceutical Company. And they, everybody that was in the biology division, I don't know how many people it was, 6, 8, 9,000 people, got the choice of joining the new pharmaceutical company or leaving, basically. You didn't get fired.
What year was this roughly?
So I went there in 1984 from molecular genetics, and so that would have been 6 years from now, would have been about 1990. And so I tried this for a while. I became a group manager. Lots of people reported to me. We did antiviral drug discovery. We did antivirals for flu, herpes simplex virus. I had a little CMV group. There was another group that was working on HIV, which was one of the most popular ones. DuPont got some drugs out of that one. Didn't get much out of me. Our viruses were tougher to discover. And I had fun with that, I guess, for a little while. But then I really realized I'm not doing anything that I like. I had a couple of postdocs funded through the NIH that I kept working on pseudorabies virus. And I said, I like that stuff better. And at that time, I had begun what is now my life's work, which is really trying to understand how these herpes viruses get into the nervous system. You know, they establish a latent infection in your peripheral nervous system, and then they can reactivate and cause lesions like cold sore sores or shingles. Nobody knew how that worked. And I got interested in the cell biology of that. And we had isolated some mutants that would spread one way in the nervous system and not others. And I started working with neurobiologists because we could use these viruses as tracers of neuronal connections. They spread in neural circuitry. And so now I'm funded by the NIH, the National Neurological Diseases and Stroke, to do work on uncovering how herpes viruses infect and spread in the nervous system, how the nervous system responds to infection. So we're learning a lot. And now I'm back where I started, which was the intellectual aspect of trying to understand how viruses do what they do.
So somewhere in the late 80s, you decided you didn't want to be at DuPont anymore.
So actually, in the late 90s, early 90s.
Early 90s.
It was early 90s.
And you had this nice herpes program.
I had.
You look for a place to bring that.
That's
basically.
right. I started looking in academia. I realized early on when I went into industry that I had to keep one foot in the academic arena. So all the time that the 14 years that I was out in industry, I was attending meetings, I was giving talks, I was on study sections, I reviewed papers for journals, I kept my academic contacts up. You know, you and I met, I think, way back at the NIH when you were first in David's lab. You were trying to do some cloning of some kind, I can't remember. But all of the people that I met during my cloning phase, you know, were out getting things set up. And so I didn't really have too much trouble finding universities that would like to have me there because, first of all, I knew something about industry, and second of all, I worked on some really serious problems that were of fundamental interest.
So you moved again, one more time.
One more time, and
that's it.
And that's it. My wife says we are here forever.
We have a lot of listeners who are really early on in their careers in science, graduate students and postdocs. So is this hard to do, to be in industry a long time and then get back into academia?
Yeah, I'd say it's particularly hard if you do typically what happens in industry. You will work on one project for a while, and then the company will either move you off it or kill it, and you have to move on to something else. So you may come in with an expertise in a particular area, and you may be using that expertise in many different places as you move down the road, and you learn new things. And as a result, you're spread very thin. And so when people look at you, they don't really know what you're good at anymore, except the company knows that you're a damn good cloner or you're a good sequencer.
You never get to develop a real program.
There are very, very, very few people develop programs. There are some that do.
Like you.
And they're exceptionally lucky. If you go to a place like Genentech or someplace that really values basic research, you may be able to continue your particularly intellectual path as you spin off products, but you have to spin off products and or ideas for products, or be seen as a generator of ideas or something like that.
Yeah, but I remember you said once the easiest thing to do in a company is to cancel something.
Oh yeah. The classic management tool is to say no, because then nothing will happen, and most of the things that happen in the pharmaceutical business don't work. And so by canceling something, you just improved your the chances for success.
On the other hand, you'll never get back into academics because you don't do anything.
Yeah. But for me, because I still maintained an image in around the academic community, and people knew me before I'd gone into industry and then afterwards. So most of the people that knew me before were now department chairs or senior faculty members. And so Arnie Levine was a good friend of mine. I'd known him for a long time, and I was always impressed with what he did and how he approached things. And he started this new department at Princeton in 1984, and he invited me to be on a member of their scientific guests or something like that. So I came up every year and I lectured in Tom Silhavi's prokaryotic genetics course on Lambda, and Tom Schenk was here, and I talked with Tom quite a bit about things. Jane Flint was here. She was an adenovirologist. And this was a community I felt very comfortable with, and they were comfortable with me. And in 1993, they offered me a full professorship, and I took it. I came in December of 93, like that.
Yeah, you kept your irons in. That was the key. You kept.
Yep.
You as you said, you were there at the beginning, and then while you were gone, you kept in. But for most people, it's very hard to do. So people who are thinking of going into industry, it's going to probably be that's their last career move. They're not going to get back.
Yeah, industry is very interesting because my experience in industry was fantastic. I have to say, I wouldn't be as happy now being here had I not been there.
Yeah.
Because first of all, I learned a lot. I learned about how companies work. They taught me an enormous amount of management skills. They sent me to courses on managing budgets and things were sort of boring at the time. But, it enables me to be a department chair, to do a lot of things with really no sweat. This is actually pretty easy. This department is rather big for a small Ivy League school. We have millions of dollars of income that come in, and for some people that's sort of scary to manage that amount of money. I just know I feel comfortable with it, and my bosses up in the administration are comfortable with me, and they taught me how to deal with different kinds of people. When you run a lab, it's a little microcosm. And you've got people, you have personalities you have to deal with. You have to motivate some people. You have to demotivate some. You have to convince people that, that's not good behavior, or it's not going to happen in my lab. And you have to be strong enough to be able to say no, and you have to be wise enough to figure out that you need to let people go and figure out what they're doing, even though you don't like what's happening, but this could be a good direction to go. All of that I think I learned in industry.
Yeah, your management skills clearly, right? Because that helps you be a chair, and a lot of scientists don't have those skills.
Yeah, well, they never teach us how to do this stuff. I now when I see assistant professors come in my department here, and right off the bat, they have to run a lab, they have to manage, grants. They have graduate students that are just learning. They have to teach. They don't teach you how to teach in most places. They throw you in the deep end of the pool. In industry, you have to learn how to speak. You have to learn how to speak to people who don't know what you're talking about, and you have to convince them that they have to like what you're doing. And they're even though some people say well, these guys don't know any science. I'm telling you, they know a lot of things much better than you do, and you can't pull the wool over their eyes. I learned got a lot of respect for people. I also discovered that there are shysters wherever you are. It's in academia or in industry. It's not a unique thing. It's just learning how to get along with people. And if you keep your passion and your joy for doing stuff, you can make a living almost anywhere. I have friends in industry that don't mind being moved around. They're very happy with what they do. In industry, it's all about teamwork. In academia, it's pretty much all about you. And you can work in teams, but when the rubber hits the road, you got to write the grant, you got to get the tenure, you got to do all of this. Whereas in academia, if you're a good team player, you may not rise up above the crowd, but you're an integral part of the team. You're going to get paid. You're going to survive. It's going to be very comfortable.
So your chair, you have the journal business, you run a lab, you raise money for it. How many people in your lab?
So we were just talking today. I've got 5 postdocs and 4 graduate students and 2 undergrads.
It's a good-sized lab.
Yeah, I don't want it to get much bigger because I still know what they do.
And you teach an involved course every year to undergraduates.
I just finished.
So you must love all this.
I love it. Yes.
What's the best part? Is there any best part, or?
Oh, gee. There's always come in the morning and a graduate student or somebody will either have a note on my door, saying, come see me right away, or I'll be leaving at night and someone, you ought to come in and see this, and see what's going on.
That's great.
Discovery.
Discovery is the best part.
It's great. Or a student comes into me depressed. They've been doing this experiment. It's not working. They can't figure out what it is. And I say, let's go to the blackboard and you tell me exactly what you did. And all of a sudden, I see what's going on. And they say, how do you know that? I said, well, I've seen this before. Yeah, and then all of a sudden they realize it's not them. They're not, it's the technique. They're not adding the right amount of salt or whatever, and it'll work. And, getting your self-confidence up to do an experiment that's complicated and to do an experiment that nobody else has ever done, so you don't really know what the results are going to be, to have confidence in the result. When graduate students come in, almost always, and they're probably right, most of the time it's because they're not doing the experiment right. But very soon they switch over to the other mode in which they really take off. That's the best.
Yeah,
that's the best.
I understand that part too. I want to read a couple of emails to you before we wrap it up. So this has been a big year for virology.
Oh, yeah.
So last fall we started with a couple of guys getting Nobel Prizes.
Yep.
Then we had a pandemic influenza.
Yep.
Now we have XMRV.
Fantastic.
So any of these particularly interesting to you? I follow them all the time.
Yeah, well, flu is always interesting to me, simply because when you're around, you teach in the fall, right, like I do virology, students begin to get respiratory diseases and whatever, and it's always fun to talk about. Now this year, with this H1N1 invasion that we're having, there's more students are sick. I don't know how serious it all is in terms of the number of students that are getting infected, whether it's the normal amount or whatever. But the university has taken extraordinary means to publicize, how to stop this from happening, how to sneeze, how to wipe your hands, and all of this stuff. And so there's a lot of interest, and so it's great for teaching. I mean, I have changed my slides and things so that I could show off what goes on. But this XMR, so xenotropic murine-like retrovirus or whatever.
It's Maloney-like, basically.
Yes, yeah.
Remember Maloney?
Yeah,
Everybody works with.
absolutely.
So this is a endogenous version, which can't infect mice. Yeah. Can only infect other cells.
Yeah. So what really got me about this one, and this is just sort of a private little thing, is when I was at DuPont, and I had now become part of DuPont, part of DuPont Merck, and one of the things that I had in my group was a small group that was involved at that time in building what is now the capillary sequencers that are all around. So I had so they wanted big things to try to sequence, but not too big. So they were sequencing herpes DNA and whatever, and we were sort of the test case. So I had a guy who was sequencing herpes DNA. And we went up to Boston where we had another site and met some people that were studying chronic fatigue syndrome, which was really hot stuff right then. It was just coming out. And it was all of a sudden the understanding that this just wasn't some sort of group of malingerers that were complaining and sleeping late or whatever. This was really some kind of syndrome. And the idea at that time was that it was caused by HHV6. It was a brand new human herpes virus that was discovered in AIDS patients. We now know that this is the virus that causes roseola in kids, and we don't know what it does late in life, but just about everybody's infected with it. It's a very common virus. So the idea was that that chronic fatigue syndrome was caused by HHV6. So we actually started sequencing HHV6, and we developed a variety of diagnostic probes and things. So I got an interest in it. I read a lot about it because it was happening. Part of it was happening in my group. And I realized that these chronic kinds of diseases, these strange sorts of syndromes, you can find a virus for anything. There's viruses out there. We're surrounded by them. We live in a sea of these guys. And if you look hard enough,
a literal cloud.
You literally can find anything. And proving it was really hard. You couldn't do Koch's postulates. You couldn't do a number of things. It was always associations. And the associations were of the kind that really were frightening to say, this is what it is. And so I lost track. I didn't pay any attention. So when this came out, I read very carefully because for the first time, I could see that people really had done the right experiments. Now, we still don't know because if you don't have chronic fatigue syndrome, I can't take the virus and infect you and then see what happens. So it's still associations. But the idea really looks right to me, is that this is probably a common kind of virus. It's infecting people at some rate. And then most of us can handle it, but there's another group that has some kind of genetic disposition, maybe multiple mutations or something, that makes them particularly susceptible for whatever this virus does to the cells that it infects. And that manifests itself as this disease. So it's like this convergence. So you have a common virus and some mutant people, and they get together and you get it. This is kind of a popular idea that some of the syndromes that you see, and you can just pick them out. I don't want to name too many, but obesity and autism and whatever, that you have something that's happening to all of us, but there's some of us that have a particular predilection, and when those two things get together, then you get this. Neither one of them is going to get it. It's when they interact. So this actually, happened with HIV. There was HHV8, human herpes virus 8, the virus that causes Kaposi sarcoma. That was an epidemic that we didn't even know about. That was going on, and when the AIDS epidemic happened, and you got AIDS, people that had the AIDS syndrome got infected with HHV8, it manifested as Kaposi sarcoma. They got sarcomas that they should have never had before. So that's a good example of, two infections going on, and then when the two get together, you get something that neither one had in common.
Well, I have always said we need to know the nature of the viruses in everybody, because this has been in a lot of people for years, and we didn't even know it. Yeah. But we've for many years been able to do microchip arrays, right? Yeah. And we should be doing it on everybody, because I bet we'd find a lot. Yeah. And actually amazed that we haven't picked this up before, because so many microarrays have been done. Yeah.
And I really don't know the sequence of this guy, so I don't know. It's probably 10,000 base pairs or something like that. It's not a very big virus. No, and I don't know if its relationship to the other retroviruses is distinctive enough so that you would be able to pick it up. I just haven't followed that.
So this is about, it's more than 90% identical to Maloney. Yeah. To the xenotropic Maloney. It doesn't change very much. And apparently the prostate samples and the chronic fatigue are, 30 changes. Yeah. And so it's very interesting that it's not evolving very much. I'm amazed that it's in so many people that are healthy. So as you said, there may be other factors.
Well, there was another virus this year. It's not as well picked up by people, but there's a very strange kind of cancer. It's a Merkel cell carcinoma.
Polyoma, right?
And that turned out to be polyoma virus. Yeah. And that was discovered actually by the team that discovered HHV8. Right. Essentially looking in diseased tissue and comparing it to normal tissue and figuring out what the difference was. And there's a new virus in there. Well, it's not so new. Turns out this thing you find it in a lot of places as people are looking for it. We just missed it.
No, we have to look a little harder. And I think we need to, NIH always has liked this hypothesis-driven research. We have a problem. But this kind of searching, fishing, it needs to be done, right?
Well, that's probably why virologists love fishing, because
that gives them a chance.
I've got this little saying here that says, the charm of fishing is that it's the pursuit of what is elusive but obtainable, a perpetual series of occasions for hope. So it's great.
Did a fisherman say that?
Yeah, it was John Buchan, 1875 to 1940, British author, statesman, and angler.
Excellent. Well, Dick will love that. Let me read you a couple of emails that we got. You might be interested in thinking about this. This one is from Jim. He says, I find your podcast one of the most informative and entertaining on the web. I would like your opinion on the following items. My niece received chickenpox vaccine age 6 and developed herpes zoster at age 12. Mm-hmm. I thought that herpes zoster is a reappearance of chickenpox virus and usually only appears later in life when a person is immunocompromised. Could this current infection be related to vaccination? My niece is in apparent good health, but I am concerned that this infection could signal the onset of a serious underlying condition such as leukemia.
Well, this is really a complicated issue, because the chickenpox vaccine, I guess for people who don't know, chickenpox is a herpes virus, and the primary disease that shows up is chickenpox, and it's a very interesting epidemiology of this virus. But it goes latent, it shuts itself off in your peripheral nervous system, and anybody who's ever infected with chickenpox has got the genome in your peripheral nervous system. It'll stay there the rest of your life as long as your immune system is good. In some patients, when they get to be 40, 50, 60 years old, the immune system seems to slow down a little bit, and you can get a reactivation of this virus. Reactivates, it comes out, and it presents itself as zoster, which is could be a rash in a specific area of the chest, the thighs, the face, and whatever. You get immunized when that happens. You get a boost, and you probably will never get zoster again. from that. So that's sort of the disease. And it can be painful, it can be itchy. There's a lot of things that are associated with that. But it generally is that you get chickenpox as a kid and zoster as an older person. Now, if your immune system goes down in the middle of your life, chemotherapy, you get AIDS, you get measles virus, you get Lyme disease, any of these things that cause your immune system either to go down or to change in some way, the virus can reactivate and you can get zoster. So the rule about age is not a good rule. It's all about the immune containment of the virus. Now, the chickenpox vaccine is a live virus. It's an attenuated virus that was first developed in Japan to treat kids with leukemia, because their immune system is really not, you have to be very, very careful. And if they ever get real chickenpox, they can die from it. And chickenpox is so common that kids really have to be protected against it. And so the Japanese scientists developed this live virus that didn't cause disease in these kids, but immunized them. So that's the basis for the vaccine. Merck develops this vaccine now, and it's really quite good. And there's two kinds of vaccines, one that's given to kids, and then there's another one that just was licensed, I think, last year, that's given to adults.
Zoster, right?
To boost you so you don't get zoster. It's the same vaccine, you just get a little larger dose of it.
Right.
So for the email question, the little girl who got this got a live vaccine, and it is conceivable. There's a number of ways to think about this. She could have had, this could be the vaccine strain coming back out. You'd have to sequence that because it is alive. It could do that. When it does it, it's very rare.
Right.
I really am not an expert on this, but I know that they can't stop it from reactivating, but because it's so attenuated, it generally is taken care of. I think, this is one of those things where you should really talk to a physician, an infectious disease doc, and get the real picture. I can tell you the molecular biology, but I can't tell you, what to do as a parent and whatever.
So my colleague Saul Silverstein said you'd have to sequence the virus to see if it's the vaccine strain.
Yep.
And he said it may be immunosuppression, but
Yep.
you should go to your physician and find out. It certainly sounds like they should go to a doctor and follow up.
I know several people in my labs. I got zoster. I'm just I'm 35 years old. What happens? It happens. And so a lot of times you go back and you discover, that they had Lyme disease, or they, had a,
right. That's a good point. Some other infection. It
Yep.
could even be a subclinical.
Absolutely,
right?
absolutely.
It would
All you may know is that you're not feeling quite right. The next thing
Yeah.
you know, you got this nasty rash.
All right. So he also asked, I've always wondered if the use of mustard gas in World War I could have influenced the 1918 pandemic. As you know, mustard gas is a mutagen, and thousands of tons of the material were used in 1917.
My goodness.
So I guess he's wondering if the mustard would have mutated the virus.
Yeah. Well, the interesting thing to me is we've now sequenced, at least from people who died in 1918, this virus, and it doesn't have a lot of mutations compared to the other flu viruses that we have around.
Yeah.
It also looked like it had segments that came from birds and whatever. This is one of those things where it's very hard to say no,
Right.
but it seems very unlikely.
Yeah.
All of those poor soldiers over there who were gassed.
Yeah.
I feel bad just thinking about it. My grandfather actually had that experience, but and he got the flu too. But he got it because he got blood poisoning over there, and they put him in Fort Riley, Kansas, which is like the epicenter of the 1918 flu.
That's where we think it might have. Well,
Yeah.
it started somewhere else in Kansas, Haskell, Kansas,
Yeah.
and then made its way up to Fort Riley.
Yep. So he probably. It might have had more to do with the fact that the incredible stress and the treatment that the soldiers got made them more susceptible to these kinds of things. We don't really know.
It could have made people die more readily, sure, but I don't think it would have had much of an influence on the virus. I mean, the flu is pretty good at changing on its own.
Yes.
It doesn't need a mutagen.
Yeah.
But also, we don't have much evidence that there were different strains arising because we only have the one that was reconstructed.
That's right.
So.
So we don't know.
Finally, when I was taking pathogens class many years ago, we argued about the most painful way to die. I always thought it would be rabies, followed by tetanus. What do you think?
Oh, God.
I think rabies is a really bad one.
Rabies is horrible. A lot of people forget about this, but rabies is one of the most dangerous viruses on the planet because without treatment, it's always almost 100% fatal.
Yes.
And it's a very slow virus as it moves into your nervous system.
Yes. I know rabies well because we always teach it and we talk about how terrible it is. But I think tetanus is also quite painful.
Is not,
but I would put rabies and tetanus.
I think so. Right up there. Yeah.
All right. One more. This is from a fellow. I am a honeybee breeder in California, and I listen to TWiV while I'm artificially inseminating queen bees. That's great. So if you're listening now, don't make any mistakes there. My work is primarily breeding bees that are resistant to the varroa mite, which as well as sucking hemolymph blood of the bees, also is a vector for virus. One of the viruses involved in the demise of bees is Israeli acute paralysis virus, which was discovered by your colleague Ann Lipkin. So I discovered TWiV in an effort to learn more about virus. Just about everything I know about virus comes from listening to your show. With an 80-hour work week during bee season, I really appreciate being able to get good educational information while my hands are otherwise engaged. Well, he's come to the right place. At least I'm a virologist and not something else. Recently, a paper in PNAS described a finding that due to multiple viral infections, unusual fragments of ribosomes have been found in the guts of bees. It's thought that with the destruction of these protein factories, the bees are unable to produce substances they need to react to threats in the environment, like pesticides and pathogens. This may leave them susceptible to almost everything and is probably a contributing factor to colony collapse disorder. My question for you is, how would a virus be involved in the destruction of ribosomes? It makes me realize how little I know about the basics of viral reproduction. Are ribosomes involved? Please continue with Virology 101, as I have a long way to go to getting a good handle on the basics. What do you think, Lynn?
So this is, there's a very easy part to this. It's one of the fundamentals of virology. All viruses have got to make messenger RNA that can be read by host ribosomes. So the ribosomes are the little factories that bind the message and make the proteins. And so viruses absolutely require ribosomes as part of their life cycle. So if they destroyed them,
you're in trouble.
They would be in trouble. I could imagine that maybe, they might reduce the efficiency of protein synthesis, but this is a dangerous game to play if you're a pathogen. Because when you begin to affect fundamental processes of the cell will kill itself. The cell is got this phenomenon, it's called homeostasis. Things have to be kept in balance, cells, tissue, and when the balance is disrupted, bad things can happen. And one of the things that happens in cells when you stop protein synthesis is the cell dies. And it dies a very interesting kind of death. It's called apoptosis. It disrupts its DNA, it makes membrane blebs, the macrophages come in and clean everything up. So I don't know this paper in PNAS, but I just know from first principles that destruction of ribosomes is likely to be a byproduct, not a primary aspect of what goes on. That's what I would.
So the idea in this paper is that, well, this is my idea, is that the viruses are probably activating the innate immune system, and part of that is an enzyme called RNA cell, which is an ribonuclease that destroys all RNAs, viral and host. The idea being you sacrifice the cell to try and just stop the infection. And one of the targets of RNA cell is the ribosomal RNA.
That's very good.
So it could be an innate response of the bees. And they didn't look at it in the paper. They just suggested.
It is interesting, that the fundamental idea about innate immunity, one of the proteins was discovered in Drosophila in an insect. The toll receptors were discovered there.
Absolutely.
And so, insects, even though they seem to be simple on the one hand, they've got the basic kind of immune system.
They have innate immunity.
Innate immunity.
Not adaptive, right? Just
no adaptive.
The situation with the colony collapse is interesting because no single virus seems to be involved. So it's a similar situation as you were talking about before. About you may have XMRV, but maybe some other virus is also contributing. So it may be, and that makes it hard to sort out in the end.
That's right.
At the end of TWiV, we usually do a pick of the week, and we pick something to recommend to listeners that we like about science. And I'm going to go first so that you can think about something. And my pick this week is a website called Viral Zone, and this is it's somewhere in Switzerland, I believe. It's a part of the organization that takes care of sequence databases over there.
SwissPro.
XPC SwissPro. So they have a web resource for all viruses and families. And what they do is they have properties, they have illustrations, and they have links to all the sequences that you can get. And I found it great. It often is the top hit when I'm looking for a picture of a virus. So it's called Viral Zone, a web resource for viruses. That's my pick of the week, and I'll put the link in the show notes. You got anything that you like?
Yeah, well, my pick is a book I'm reading. In fact, I just finished it because I couldn't put it down. It's a biography of Francis Crick. And it's by Robert Olby, who really writes these kinds of science things well. It's called Francis Crick, Hunter of Life's Secrets.
Great.
That's his name. And Crick is an amazing guy because of how he made such fundamental discoveries. He worked with Watson to solve come up with a structure for DNA. He and Watson have a paper, which I was just reading because I'm going to talk about it in class. It's called The Structure of Small Viruses, March 10th, 1956.
I remember that paper.
That's right.
And
the first sentence, it is a striking fact that almost all small viruses are either rods or spheres. And, of course, they didn't know very many viruses.
Sure.
And the only ones they talk about in this paper are all plant viruses. But they come up with the idea that the rods are all helical symmetry and the round guys are all icosahedral symmetry, or at least platonic solid symmetry.
And they were right.
And they were right. It's a very short paper. And so he's done this time and time again. And now I'm really interested in neuroscience and viruses that infect the nervous system. And Crick, later in his life, moved to study the problem of consciousness and whatever. And one of my, I can't say that he's a colleague. We've never really done that many things together, but somebody that I really admire, Sidney Brenner. And Brenner and Crick, came up, they through genetics, they figured out that the code was a triplet code. They figured out there were things like transfer RNA. They figured out there was messenger RNA. I just had to read this book because Olby is really a great author. And so I highly recommend it. It's really a treat to read.
Francis Crick sounds great. I'll put it on my list. All right, thanks a lot there. Well, that should do it for another TWiV. If you're a new listener, please go to iTunes and subscribe. And if you like it, leave some comments. That really helps us. And if you don't use iTunes, go to our website. It's twiv.tv. We keep all the show notes there. And if you like TWiV, tell others about it. That really helps us spread the word about viruses. So we're at twiv.tv, and you can send us your questions, comments, criticisms. Tell us if we got something wrong. You can send us an email to twiv@twiv.tv. You can send us an audio file if you'd like, an MP3, or you can call up Skype and leave a message. Our name is TWiV Podcast. You have about 10 minutes if you'd like. Or you can tag storiesanddelicious.com with 4 TWiV podcasts, and that sends them to us if you have something that you'd like us to talk about. Lynn, thanks so much for spending time.
Good to see you again.
I loved hearing that history. There's a lot of good stuff in there. And you've come a long way, and you have gone through the whole history of virology, except for the 50s, which is really when it started. Appreciate your time.
Great. It's my pleasure.
And I hope the kids in your class listen to this.
I hope they do, too. I know my postdocs listen to it. They're always telling me how they're listening to it.
I told your class, so I taught your class a couple of weeks ago, and I told them to look out for this episode. And in closing, we should probably note that you, Ann Skalka, and Jane Flint, all of whom were mentioned, and myself all wrote a textbook.
That's right. Everything we know about virology.
That's right. So it's a small world in the end. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
Oh, yeah.